All right, are there any questions from last time? Any questions? So last time we introduced the cell, right? Then we were looking at the review of organic chemistry, right? And we covered these different kind of compounds, and we had talked about amino groups, and then we talked about how an organic acid can react with an alcohol, all right, to produce an ester and water. Okay, we looked at that. And then I started basically talking about phosphates. Okay, phosphates are also very important in in, bio, in biology, and they play an important role. And I will talk about that in a few minutes. All right, what the important role is. So before that, phosphates are derived from inorganic phosphoric acid, that is H3PO4, right? If you remove two hydrogen ions from it, right, you get the phosphate ion. Okay. Now the phosphate ion can react with a number of other ions and molecules in different ways, and that's what we are going to look at on the following slides. Right? The inorganic phosphate ion can react with an alcohol, right, to produce basically what is called a phosphate ester. Okay. Again, this is like a salt, alcohol reacting with an acid. This is not an organic acid, but it's still an acid. Okay, so it's a phosphate ester bond, the bond that you see over there in the shaded blue region, right? And a water molecule is removed. These phosphate ester bonds are very important in the formation of nucleic acids such as DNA and RNA. Okay, in fact, like DNA is made up of different units, right? And the units are linked to each other by what are called phosphodiester bonds. So two phosphoester bonds basically link up the individual units of DNA or RNA to form the respective nucleic acid, right? And I will elaborate on that, probably within the next 30 minutes, 30, 40 minutes or so. Okay. So this is important. Then another example of a phosphate ion reaction is a phosphate, right? The inorganic phosphate ion reacting with a carboxylic acid, right, to produce what is called a carboxylic phosphoric acid anhydride. Okay. Why is it called anhydride? Because this is formed by the removal of a water molecule, right? So you're taking water away, so it's called an anhydride, right? Then this particular reaction is extremely important. Okay. So two inorganic phosphate ions, right, can react with each other, right, with the removal of a water molecule. So you see those two hydroxyl groups in the middle, right? Between the two of them, you remove a water molecule, and you form a new bond, okay, between these two inorganic phosphate ions, and that is called a phosphoanhydride bond. Okay, again, anhydride because it is formed by the removal of a water molecule, right? And this is a high energy bond, okay, and this is actually the bond in which the energy in all our cells is stored. Okay, and let me just switch a little bit from this to actually writing on the screen, because I want to show you how energy is stored in our cells. Okay, in order to store energy, you will form a new phosphoanhydride bond, right? In order to use up energy, you will break phosphoanhydride bonds. Right? We are going to encounter this again in the next chapter, right? But I just want to introduce it right now, right? So let's just see if this thing works, all right? So the molecule using which energy is stored in our cells is called ATP, right? It's adenosine triphosphate, right? Right. So you have some molecule A, right? We'll worry about what that molecule is, okay? That actually is linked to one phosphate group here, for one inorganic phosphate ion, then another one here, right? And then another one here. So it's triphosphate because you have A linked to three of these phosphates, okay? Now these bonds between these individual phosphates, okay, they are high energy bonds, okay? So if you have A with only one P, then that's called AMP or adenosine monophosphate. So this is ATP, right? With only one with only one phosphate in there, it's adenosine monophosphate or AMP. If you have two phosphates in there, that's adenosine diphosphate or ADP. Okay? If you have three phosphates in there, that's adenosine triphosphate or ATP. Okay? So in the cell, all right, you have both ATP, you have ATP, you have ADP, you have AMP in the cell. Okay, so whenever you want to store energy, okay, typically you will start with an ADP, that means with a diphosphate, you will add the one extra phosphoanhydride bond and form a triphosphate, right? So you have stored the energy in this bond here, right? So when energy is needed, so these molecules, they can move about in the cell. Okay, so it's like energy, you have mobile energy carriers, all right? So when energy is needed, then it is going to break that phosphoanhydride bond, right? And go from ATP to ADP, okay? This is called the ATP-ADP cycle, right? So this is the ATP-ADP cycle. And this is the mechanism by which our cells are powered, okay? So that is like the currency of the cell, okay? So that's why I said this phosphoanhydride bond is extremely, extremely important, right? And in many of the reactions, okay, it's, yeah, just one moment. It's just like, you know, if you want to buy food from the grocery store, you have to go with money, okay? You want to buy gas, you have to pay money. So you can think of this like the currency of the cell. Yeah, what was your question? No, you probably can. You probably can. Okay, but that's how nature has figured it out. Okay, why did it select A? I don't know, right? But this is the mechanism by which all the cells are powered. Okay, yeah. So, and we will revisit this. This is so important that in the next chapter, when we talk about energy considerations in biochemical reactions, okay, we will revisit this. But I wanted to introduce it right now, right? So that it's reinforced in your mind. Because even if you go over the lecture from last time, okay, you can see that there are a lot of things you have to remember, right? And I told you that it's not going to be easy, right? That, but I think it's going to be a worthwhile experience because I'm trying to walk you through this, picking up all the important things, okay? Because each of those books, the book from which, you know, I got all notes for all this stuff, that is like 600-700 pages. And the other book on plant physiology is also 600-700 pages, okay? So, I mean, not everything is terribly important. We want the big picture, right? A good working knowledge. Okay, any questions on this before I switch over to the slides again? Okay, so I impressed upon you the importance of phosphoanhydride bonds. Okay, and you will see that many times in this course. Next, we move on to the building blocks for common organic molecules, right? Now, as I mentioned earlier, right, although you have so many different kind of organisms, right, but the building blocks, right, for most living things are the same, okay? The proteins are made up of the same kind of molecules, okay, units that are strung together and so on. So that makes the study a lot easier, okay? So here we are going to look at the basic building blocks. Now, in uh, in living organisms, all right, one difference between organic chemistry and inorganic chemistry is that in living organisms, the molecules are huge. Like in inorganic chemistry, you'll have like sodium chloride, okay? It's a pretty simple molecule, okay, low molecular weight. In organic chemistry, you know, you could have, you know, weight could be in millions, right? Millions of daltons, okay? But the good news is that these huge molecules, are what are called macromolecules, they are made up of simpler modules, okay? So if we study the simpler modules and look at their properties and all that, we can readily gain an understanding of what the big molecule is going to do, right? So it's quite systematic that way. So there are 4 classes of these small organic molecules or modular units that are combined together to form the large macromolecules. The first one among them are the sugars, which are the building blocks for the more complex sugars and carbohydrates. Okay, you hear that all the time, but at least in this course you will get a feel for what it really is. Okay, then you have molecules that are called fatty acids, which are the building blocks for. Yeah, yes, sir. Go ahead. Yeah. It's possible. I just don't know the answer to your question. It is? Okay, 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 okay. Yeah. That's possible because a lot of things in nature, they happen that way, you know. So, yeah, that's possible. Well, I haven't done the energy study and all that, you know, to be able to answer that. But if you've heard it in some other class, probably that's true. Yeah. Then you have fatty acids, which are the building blocks for fats, lipids, and all the cell membranes. All right. And I talked a little bit about this before, right? Remember? I mean, we talked about the lipids and the, you know, hydrophobic part, hydrophilic part, and all that. Okay. You will see how that comes about naturally. Okay. In the case of fatty acids. Then we will also discuss amino acids, which are the building blocks from which all proteins are made, right? And then the last set of building blocks that we will look at are what are called nucleotides, which are the building blocks for nucleic acids such as DNA and RNA, right? Again, I mean, by the time I finish today's lecture, you will know what DNA is, what RNA is, and so on. So first, let's look at sugars, right? So the simplest sugars, which are called monosaccharides, are compounds that have the general formula CH2O, right, and N, right? So that's why it's called carbohydrates, okay, because it's carbon and water, okay, CH2O, right, and N can be a number that is usually like 3, 4, 5, 6, or 7, okay. If N is 6, then it's the sugar that is called glucose, right. So monosaccharides usually occur as aldehydes or ketones, okay. So what are aldehydes? What is the defining property of an aldehyde and a ketone? Not you, okay, you had their biology before, so I want to see you, other people in the class, What was the defining property for an aldehyde? Anybody remember? What was the group that would define an aldehyde? Again, huh? Yeah, C double bonded with O and then H, yeah. Okay. I mean, even if you didn't pick it up in one class, that's fine, keep reading it, right? You'll pick it up. And what about the ketone? Ketone is characterized by the presence of the what group? Yeah. So it's called the carbonyl group. Yeah. It's all there in your notes. So go ahead and read that again and again. No homework doesn't mean that no work at home. Okay. There is work. Right. So monosaccharides usually occur as aldehydes or ketones, and we give a few examples. All right. We'll look at 5 carbon sugars and 6 carbon sugars, okay, because they are extremely important in molecular biology, right? 5 carbon sugars are called pentoses, right? And 2 examples of that are ribose, right? Which is an aldehyde, right? Again, remember at the top you can see that C double bonded with O and H, right? So this is an aldehyde. It's a 5 carbon sugar. It has the general formula CH2O and N is 5, okay? Or it could be a ketone. On the right hand side, the ketone is ribulose. You can see the carbonyl group in the middle, C double bonded with O, and then 2 other groups on either side. Okay. Now one more thing that I want to point out. So after this 5 carbon sugar, we will talk about 6 carbon sugars. But here what I want to point out is that usually the carbon atoms, all right, they are numbered from top to bottom, right? That means starting from the aldehyde group, you number them down. So you will have C1, all right, C2, C3, C4, and C5. Okay, keep that in mind. Now, one more thing is that in aqueous solution, right, when you put this molecule in water, you will never see this structure in any book, right? The structure is going to be different because in aqueous solution, what is going to happen is that this last guy here at the bottom, right, CH2OH, right, that's like a separate, that'll become like a separate unit that'll stick out of the plane, right? And then the first guy here, the carbon number 1, right, that oxygen will bond to carbon number 4, right? I'm going to draw the diagram and show it to you in a minute, right? But I'm just telling you what's coming. It'll bond. So the carbon number 1, the oxygen, is going to serve as the bond between carbon number 1 and carbon number 4 here, right? The CH2OH will stick out of the plane of the paper, right? It's just sticking out. Everything else is in the same plane, right? That's the kind of molecule that you will see, right? And I'll show you just in a few minutes, okay, what, I mean, or give you an actual demonstration because I don't have it in my slides, okay? But that's usually the structure that you will see for this sugar ribose. If you open a book, let's say, a medical book or some other book, okay, they're not going to have this structure here because usually inside the cell it's an aqueous environment, okay? So the carbon number 1 will form a bond with carbon number 4 using that oxygen. The hydroxyl group will go into carbon number 1, right? And you will have a ring structure that will be formed. And the same is true even for the 6 carbon sugar, which I have on the next slide, right? So the 6 carbon sugar is it's a hexose, all right? It has 6 carbons, all right? And if it's an aldehyde, again, C double bonded with OH, you can see at the top, right? That's an aldehyde. That is glucose. So glucose is an aldehyde. On the other hand, fructose is a ketone, right? Right. So fructose is a ketone. So again, you can see the carbonyl group in the middle, C double bonded with O, right? And then groups on either side. Here also is the same story. You are going to number the carbon atoms from top to bottom, right? 1, 2, 3, 4, 5, 6 carbons, right? And in aqueous solution, carbon number 1 is going to form a bond through the oxygen with which carbon number? Number 5 here. CH2OH is going to stick out of the plane, right? I'll show you the picture in a moment, all right? And that is the structure for glucose that you will see in medical and other molecular biology textbooks, right? You're not going to see this structure because usually it is going to be in aqueous solution. All right, any questions? Yeah, it'll form a ring compound, you know. Let's do one of those, okay? Let me just draw it out, right? And show you, okay? So let's again switch back and let me show you. So let's do the one for glucose, all right? So you have like C, 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 and C. Okay, so this is number 1, this is 2, 3, 4, 5, 6, all right? Right? So now, again, I'm not drawing the rest of the diagram, you know, because I'm not a great artist, but what I'm telling you is what is going to happen. So this is 6, C6, okay? So this whole module here is like CH2OH, right? Whatever the hydrogen and oxygens that were sticking out over there at the bottom, right? That's CH2OH. That'll stick out of the plane, okay? But this one, through an oxygen, it'll get linked up here, okay? And whatever hydrogen or hydroxyl is here, that'll get transferred here. Right. I will show you the actual diagram in a few slides, you know. So that is going to be like a ring compound. It's forming a ring, right, with that CH2OH sticking out, right. So if I show you this one first, that way you'll be able to understand a lot better, right. It makes sense, right, when I show you the full-fledged diagram in the slides. Any questions? So the 6 carbon sugars are called hexoses, right? And examples of that are glucose, which is an aldehyde, fructose, which is a ketone, right? And glucose, you already know that, you know, glucose gives you energy, right? I mean, people take glucose if they have to run a race or something like that, you know. So the 5 carbon sugar ribose and its derivative deoxyribose, and I will explain in a few minutes, you know, how you get to deoxyribose from ribose. They are important constituents of nucleic acids such as DNA and RNA. DNA is the abbreviation for deoxyribonucleic acid, right? So the sugar in DNA is deoxyribose, right? And I'll tell you how you get to deoxyribose from ribose, right? And RNA is ribonucleic acid, right? And the sugar in RNA is ribose. Okay, I already showed you the structure of ribose, right? And what it is going to be in aqueous solution, right? Deoxyribose, what do you think this is going to be? Basically, you have ribose, right? You take away some oxygen from that, you'll get deoxyribose, right? And I'll show you the detailed structure. Now, the 6 carbon sugar glucose that I showed you serves as an important source of energy, right? You know that, okay? I mean, like, you don't have to take a course on molecular biology, right? People might have told you before they run a race, they're going to have some glucose and then run. Because it's readily absorbed. Even the other sugars and all, you know, our digestive system has to break it up into glucose before it is finally absorbed through the gastrointestinal tract. Right? Okay, so we come to the favorite ring formation that I mentioned earlier, right? So here you can see on the left you have ribose, and the carbon atoms are numbered like 1, 2, 3, 4, and then 5. I mentioned to you that CH2OH is going to stick out of the plane of the paper, right? If this is the rest of the molecule, right? In the plane of that screen, CH2OH is going to stick out, right? So in aqueous solution, the aldehyde or ketone group of a sugar molecule tends to react with the hydroxyl group of the same molecule, right? And typically it's the at least in the case of ribose, glucose is the penultimate carbon that it will react with, right? And so this is the structure that is going to be shown for ribose and also for glucose, right? If you have 6 carbons, the carbon number 1 is going to get attached to carbon number 5 through an oxygen atom, right? And then CH2OH is going to stick out, right? And this heavy shading at the bottom, that indicates that C1, in the case of glucose, C1 through C5, they are in the same plane, and then the CH2OH is sticking out, right? So since the environment inside a cell is aqueous, emphasized that last time. Inside the cell, it's a watery environment, except for all the membranes for the different compartments, right? Which are kind of oily, made up of lipids. So since the environment is watery, typically glucose inside a cell is going to be shown, the structure of glucose is going to be shown like this, right? It will not be the structure that I showed you earlier with the, aldehyde sticking out at the top. So this is a structure that you're going to encounter over and over again in this course, and even if you take a course in medical school or whatever. Okay. Any questions?
So if you ask a question, what's the structure of ribose in exam? You have to remember the orientation of OH groups on
Well, I mean, I'm not going to hold. If you can remember, that's fine, but you are not a, biology student, So probably people remember that, okay, because I have not checked, okay. If I ask, I don't know what the structure is, which one is first H and O. I'm not an MD, right? I don't want to become one. Uh, Try to see if you can get it, okay. I'm not holding you accountable, okay. Just like, see, last time you were holding me accountable, okay, about that thing, right? Here, this is at least more consequential because if you're an MD, and the reason why it is consequential, I'll tell you, because at carbon number 2, if you interchange the hydrogen and hydroxyl group, you get a different sugar. That's called mannose, okay.
because I remember from my 12th grade, it was my marks were deducted for this.
Okay, so it will be deducted now also because you learned it once before and you're learning it again, okay, so you have to get it. No, I'm just kidding, but yeah, strictly speaking, like, because it's a different sugar, right? Then carbon number 4, if you do that, it's going to be galactose, right? So, but again, I mean, we are engineers, okay? At the end of the day, we have to understand that we are engineers, so we cannot nitpick on, because this is well outside your comfort zone, okay? I'm aware of that. It's outside my comfort zone too, so I want the big picture, okay? I'm not interested in learning about all the different isomers that they have and all that. Maybe when I retire, I'll do that, but right now I have other things to do, so, okay. But yeah, if you're a doctor or somebody, then probably you need to know, right? Okay, any other questions? So deoxyribose, how do you get that? So you start with ribose, which has the structure that is shown on the left, right? And then at carbon number 2, if you take away the oxygen, right, from the hydroxyl group, right, then you get the sugar that is called deoxyribose. And this is the sugar that is present in DNA. Okay. DNA is deoxyribonucleic acid, right? So it has the sugar deoxyribose, right, and which has this structure. Now, usually the carbon atoms that you see in ribose, and ribose is the sugar that you'd find in RNA or ribonucleic acid, right? I will talk about this before we get to the end of this lecture, okay? But ribose is for ribonucleic acid or RNA. Deoxyribose is for deoxyribonucleic acid or DNA, okay? Now, usually, right, when the structure of ribose or deoxyribose is indicated, the carbon atoms are not numbered as 1, 2, 3, 4, 5, right? They are now numbered as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime, okay? The reason for it is that when you look at the unit that makes up DNA, right, it's not, I mean, it has 3 things. It has an inorganic phosphate ion, okay? It has a sugar. In the case of DNA, it's deoxyribose. In the case of RNA, it's ribose, right? And then it has something, a nitrogen containing compound that is called a base, okay? So there are 3 things over there. Now the 1, 2, 3, 4 and so on without the primes is used up in labeling the carbon atoms, all right, in the nitrogen containing compound, okay? Because you have a ring compound over there, you label it 1, 2, 3, 4, 5. So you cannot label these as 1, 2, 3, 4, 5 anymore. That's why you'll see a prime on them. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, okay? Any questions? And I will get to the structure of DNA before this lecture is over, right? So you will see how different things tie together. Everything fits in quite well. So this is ribose, this is deoxyribose. You took away the oxygen from the hydroxyl group at carbon number 2, right? And you got deoxyribose. Any questions so far? All right, so now we need to talk a little bit about isomers because in inorganic chemistry, all right, once you get the chemical formula of a compound, that determines a unique structural formula and associated properties. In organic chemistry, that's not the case, okay? You can have a chemical formula, right, and you can have many different, compounds that fit that same chemical formula with completely different properties, okay? And these are called isomers. So compounds which have got the same chemical formula but different structural formulae are called isomers of each other. And organic chemistry is filled with instances of isomers whose different properties make them especially well suited for particular biological functions, right? So like, for example, if you're given a different isomer, right, to process, if your body is given a different isomer, right, it will need an enzyme called an isomerase to get it to the right form. right? If I give you a sugar in a different form, right, that you cannot metabolize, right, you have to have that enzyme. If you have that enzyme, you'll survive, okay? If you don't, you'll die, okay? It's as simple as that, okay? So, because the isomers have completely different properties, okay? And even among animals also, they have, for example, cows and cattle, right? They have different enzymes in their stomach, right? And in their gut. So they can, eat grass and survive, okay? We cannot, okay? If we eat that, we'll die, okay? We don't know how, we don't have enzymes to go and break down all the cellulose and all that stuff in the grass, right? So, but it is important to keep in mind that even if you have the same chemical formula, right, the structural formula might be different, right, and therefore the reactions that that particular molecule can undergo will be quite different, right. And to go from one to the other, you might need some enzymes that are called isomerases. So now we get back to your question, all right. So we had the structure of glucose. If you change the carbon in glucose at carbon number 4, if you interchange the hydrogen and hydroxyl, okay, then you get the sugar that is called galactose, right? If you interchange it at position number 2, you will get mannose. Yeah. Okay. So that's the reason. So there was a reason why your instructor took off points. So, but again, I mean, see, at that time, I think there was the rationale was because at that time you were probably in high school, right? So you're not specialized. Whatever is taught to you, you have to know that. Okay. Now you're doing a PhD in engineering, so it's a little bit different story. So the same standards may not apply. All right, so monosaccharides are the simplest sugars, okay? So you can link two monosaccharides together to get what is called a disaccharide, right? And the process can continue. So if you have like two monosaccharides, okay? Remember at every carbon location you have hydrogen and hydroxyl sticking out, okay? So in particular in the center, right, on two sides of this plus sign you have those two hydroxyl groups, all right? Between them you can eliminate a water molecule and link up the two units through an oxygen atom, right? And you now have a disaccharide, right? So two units linked together will give you a disaccharide. Now guess what? This linkage here can be further elongated, okay? Because you again at each of these carbons you have hydrogen and hydroxyl, okay? So you can have other units that link up, and they don't have to necessarily link up in a linear fashion, right? Because there are hydrogen and hydroxyls available all over the place, okay? So you could have branches, okay? And this reaction for linking up these two units, that's called a condensation reaction because it occurs with the removal of a water molecule, right? So in order to get the disaccharide from those two monosaccharides, a molecule of water was removed. That is a condensation reaction. The reverse reaction is called hydrolysis, which is splitting with water, right? So if you take a disaccharide and you add water to it, right, you can split it up and get those two monosaccharides, okay? So that's what happens when we digest carbohydrates, okay? Because they have to be finely broken down into glucose units before our digestive system can absorb, right? So we're supposed to eat the complex carbohydrates because they're better, okay? They give a more steady supply of energy. They don't cause your blood sugar to spikes. Of course, at your age, you guys don't worry about any of that. But uh, but finally, what you absorb is glucose, right? So you have to break it down, okay? Now the question is, this is a reaction where, you remove water, right? You get the units to stick together, right? And form disaccharides, polysaccharides, and so on. But then you add water, it'll break down, okay? So the question is, in which direction is the reaction going to go, right? That's the question that we will try to answer in the following chapter, okay? What decides whether the two units will get linked together, right? Or the units will break up and give you the monosaccharides. Okay, so that's an important question that needs to be answered. So this is an example of a disaccharide, and since there are many hydroxyl groups on each monosaccharide, two monosaccharides can link together in many different ways, and due to the availability of additional hydroxyl groups on the disaccharide, more monosaccharides can get linked to it, producing chains and branches of various lengths. So if you have short chains, they are called oligosaccharides, while long chains are called polysaccharides. Okay. So don't ask me, what is the critical number which, tells you that you're going from short to long chains, because I don't know the answer to that question. That might be the next question that I'll get asked. Okay. But I think that if you look up on the internet, maybe that answer is there. So an example of a polysaccharide is what is called glycogen, which is made up entirely of glucose units that are linked together. Okay, that's how our body stores glucose, right, in the form of glycogen. But there's a lot of water in it. Fats provide a much better storage of energy. Okay, weight for weight. I mean, they like when you burn fat, a lot more energy is released as opposed to just burning oxidizing glucose. Okay, so that is compact energy storage, right? So initially, like let's say if your blood glucose is high, you're eating too much. Okay, you don't need that much energy. Initially, your energy will be stored as glycogen units. Okay, so it is basically made up of glucose units that are linked together, and glycogen probably supplies about a day's reserve of energy, right? And after that, it is because otherwise we'd have to be much bigger. Okay, because if you're going to store all energy in the form of glycogen, you probably have to going to be 2 or 3 times your volume, right? So that's not the case. So first, for about a day's worth of energy is stored in the form of glycogen, right? And then if you still keep on eating, you don't need that much energy, right? But you're a voracious eater, right? You're in a feasting, what they call a feasting state. Then it'll start producing fats, okay? So glycogen serves as an energy storage in animals. Now the reaction that I showed you, the condensation reaction whereby two monosaccharides are linked together by the removal of a water molecule, that kind of reaction is not unique to just sugars. In fact, they are used to form proteins from their modular units that are called amino acids. I'll show you how that happens. And also form nucleic acids like DNA and RNA from the modular units that are called nucleotides. And the reverse reaction of condensation, as I said, is called hydrolysis, splitting with water, right? And plays an important role in our digestion of carbohydrates and other foods. And some specific examples of disaccharides resulting from linking together two monosaccharide units. You can link together glucose plus glucose, of course in a special fashion, right? Because I said you can produce different compounds by linking at different locations, all right? So in a special fashion, we engineers don't need to know where exactly that's linked, okay? But I'm saying glucose plus glucose can give you maltose, right? Then glucose plus galactose, right, gives you lactose, which is the sugar that is found in milk, right? Then glucose plus fructose is going to give you sucrose, right, which is one of the sugars that is found in plants, okay, in fruits and all that, okay? Any questions? Okay, so we finished sugars. Now we're going to talk about fatty acids, right? So what's a fatty acid? A fatty acid is a molecule that has got two distinct regions, okay? It is going to have a long hydrocarbon chain, right? Remember, we said hydrocarbons are hydrophobic, right? Because carbon and hydrogen, they both have roughly the same affinity for electrons, not a whole lot. So, they don't form polar molecules. So a hydrocarbon typically will be hydrophobic. It'll hate water, try to stay away from water. So a fatty acid has got a long hydrocarbon chain, and it has got a carboxyl group also, okay, which behaves as a carboxylic acid, right, and is ionized in solution. That means it produces hydrogen ions in solution, just like any carboxylic acid, and is extremely hydrophilic, right? So if you look at a typical fatty acid, right, as we show here, right, on the right you have this long hydrocarbon tail, which is hydrophobic, right, made up of only carbon and hydrogen atoms, right? These are the carbons that are shown at the corners of each of these triangles. You have the carbon atoms and then you have the hydrogen sticking out. So that's a hydrocarbon tail. And then you have the head, which is hydrophilic, right? It has got a carboxylic group. Okay. So examples of fatty acids would be like palmitic acid, which has 16 carbon atoms, right? And these carbon atoms are linked to each other only by single bonds, okay? So it is called a saturated fatty acid. In a saturated fatty acid, these are bad for you. Like the doctors will tell you that you're supposed to not ingest too much of saturated fatty acids, okay? Try to have more of unsaturated ones because they will, if you ingest those, they will tend to elevate your serum cholesterol. Right? So if you have 16 carbon atoms in that fatty acid, it's called palmitic acid, and that's the kind of fatty acid that you find in coconuts, right? That's why usually vegetables are not bad for you, in terms of cholesterol numbers, right? But coconut is bad, okay, because it has got this saturated fatty acid, right? So even if you're a vegetarian, you're having coconuts all the time, right? Your numbers can go up. Um, So saturated fatty acids are characterized by the absence of double bonds between the carbon atoms. Right. So next we move on. So any questions so far? Next we move on to another acid, which is not a saturated fatty acid. It's called oleic acid. Right. It has got 18 carbon atoms. Right. And you can see that there is a double bond in the middle, somewhere in the middle. Right. Uh, so so oleic acid is found in what? Anybody? No, again, not you. Right. It's found in olives. Right. Olive oil. Right. Oleic acid is found there. But this is, so the olive oil is going to be better for your consumption than coconut oil, okay? Because this is an unsaturated acid. But again, I mean, the presence of this double bond here is going to create steric hindrance, right? Remember I talked about last time that if you have double bonds, you cannot rotate the adjoining atoms relative to each other, right, without encountering hindrance. So this creates steric hindrance, and that is an important factor in determining the final shape of the organic molecule, right? And again, the list goes on. I don't have the exhaustive list here with me. I didn't care to learn it, right? I don't think you need to care to learn it either. Okay. If you are working, let's say, on just oils or something like that, then I mean, that stuff is available. Okay. I mean, it's been around for many years. Okay, and it's there on the internet, or you can look at books and find out. But here we only need to know that you have saturated acids, unsaturated acids, that a fatty acid has got a hydrophilic head, right, and a hydrophobic tail, right. So that's why if you put like a fatty acid in water, okay, let's say you try to mix up olive oil with water, okay, what's going to happen? The two separate, okay. Right. This is not a lipid bilayer, but they're separating from each other because, the hydrophilic part will stay in contact with the water, the hydrophobic part will stay away from it. Okay. Now, fatty acids serve as concentrated food reserves in cells, as they can be broken down to produce about 6 times as much usable energy weight for weight as glucose. Right? Remember I said earlier that fatty acids provide compact storage, right? Compact energy storage. Right? Glucose doesn't. Right? Now, usually in cells, fatty acids are stored as what are called triacyl glycerol molecules, right? Which consist of 3 fatty acids joined to a glycerol backbone. So the structure that you see on the left is a glycerol. Okay, it's like an alcohol. You can see those 3 OH groups, all right? Now, at each of those OH groups, you basically have a fatty acid that gets inserted. It's like an alcohol reacting with the carboxylic group of an acid, right, to produce an ester. Okay, so at each of those locations, so you have fatty acid number 1, which has got the side chain R1. Okay, the carboxyl group is the same for all the fatty acids. C double bonded with O, then OH. Okay, and it's the hydrocarbon portion that is different, right? So in the first case, you have R1 is the group for the hydrocarbon, right? Second one, R2 is the group. Third one, R3 is the group, right? So you have 3 fatty acids that got attached to a glycerol molecule, right? And this has produced what is called triacyl glycerol, right? Or if you go to the doctors and they do blood work on you, then they'll call that triglycerides, right? Triglyceride numbers, right? So that's how it is stored. So if you look at meat and there is, that white stuff, that fat sticking, those are triglycerides, okay? That's the actual fat that you see. Any questions? But if you look at this, okay, this is not going to be good enough, right, for making your cell membrane, right? I mean, because what you've done is, in all the locations you've put fatty acids, right? So typically for the cell membranes, in 2 of the locations you will have fatty acids. So that portion is hydrophobic, right? In the other one you will have something that is hydrophilic, right? Usually some phosphorus or some sugar or something like that. Remember in the diagram that I showed you last time, okay, I had those molecules with a hydrophilic head, and then it had 2 tails, okay. I told you that I'm going to show you why it has got 2 tails, okay. Because 2 of these locations are going to be taken up by fatty acids, right? So they are going to be hydrophobic, and the other one is going to accommodate like probably a sugar or some phosphate group, right? Some hydrophilic molecule. So that's how you'll have these units, right, for making up cell membranes that have a hydrophilic head and then 2 hydrophobic tails. Okay, that's where we are going now. So phospholipids are the major constituents of cell membranes, right? So in phospholipids, 2 of the hydroxyl groups in glycerol are linked to fatty acids. So those are hydrophobic, while the third hydroxyl group is linked to phosphoric acid or inorganic phosphate, right? And that gives you a typical phospholipid. And you can also have what are called glycolipids, where it's linked to a sugar, because sugar also likes water. Okay. So because of these properties of fatty acids and of these phospholipids and glycolipids, right? If you scatter a bunch of these molecules, the fatty acid molecules on water, right? You're going to form a layer like that, right? The hydrophobic part is going to stay away from the water. Hydrophilic part will stay in contact with the water. Similarly, you can have bubbles, okay, where the portion that likes water is going to face the water, right? And the portion that hates water is going to stay away from the water. So you can have these kind of bubble kind of thing that's called a micelle. Okay? So because of similar reasons, phospholipids and glycolipids, the lipids that are made up of 2 fatty acids for the hydrophobic part and 1 or more sugar residues for the hydrophilic part, they form these self-sealing lipid bilayers, which are the basis of all cell membranes, right? And I showed that to you last time, right? Even without covering all this stuff in detail. And I said that we are going to make the connections later. Okay. So this is the lipid bilayer, right? And I also talked last time about vesicular transport, okay? How you can take something aqueous, right? Encapsulate it inside a lipid bilayer, make contact with the membrane, right? Let the lipid bilayers merge and then release the stuff inside, okay? And do the same thing for other compartments inside the cell. Any questions? Okay, so we covered sugars, we covered fatty acids, right, and lipids, right? So that was quite easy, right? And the big molecules in the cell are going to be made up of units like this and the others that we are going to discuss. So the next set of molecules that we move on to are the proteins. Proteins are made up of units that are called amino acids, okay, that are again linked together, right? So amino acids are the subunits of proteins. They possess both a carboxylic acid group and an amino group. That's why it's called amino acid. Amino group, recall, is NH2, right? Carboxylic acid group is C double bonded with O, OH, right? So the amino group and the carboxylic acid group, they are both linked to a single carbon atom, which is called the alpha carbon, which is shown in this figure here. The carbon atom in the center is the alpha carbon, right? On the left, you have the amino group NH2. On the right, you have the carboxyl group C double bonded with OH, right? And then on the top, you have a hydrogen atom, right? And at the bottom, you have the side chain, okay? So if you're looking at different amino acids, the thing that will be different between them is just the side chain, okay? Everything else will be the same. So in nature, there are only 20 different side chains that have been found, okay? Any protein that you take, you break it up into amino acids, that R will be one of those 20, okay? So there are different amino acids that are linked together. But you can see, I mean, you're all quantitative folks, right? Like if at each location there is 20, there are 20 possibilities, okay? And if you have something that is 100 units long, right? So you have 20 to the power of 100 different possible proteins, okay? Not all of them are going to be useful because proteins, again, the function is determined by the shape, okay? If this molecule with that particular sequence of amino acids does not adopt a unique functional shape, that's not useful for biological purposes, okay? And nature would probably eliminate it through the process of evolution, right? So the proteins that we see today, they have exactly the correct shape for their function. For example, hemoglobin in the blood is supposed to bind oxygen. It has got exactly the right sequence of amino acids where it can accommodate an oxygen atom, right? Or oxygen molecule, pick it up from the lungs, deliver it to the tissues. If you go and mess up even one of those amino acids, then you have the disease that is known as sickle cell anemia. Okay. So it's very, very important. Okay. So this is a typical diagram of an amino acid, and keep this in mind, there are 20 different possible side chains. Okay. And again, I mean, like I said last time, if there's life on another planet and if it uses this amino acid kind of module, it might have used the 21st one. Okay, we don't know, right? Or an entirely different set of amino acids. So now I'll talk a little bit about the side chains. Again, you don't have to memorize, okay? Because there are 20 different amino acids, so that means there'll be 20 different names, okay? 20 different properties, right? I'm going to list them depending on your level of interest. You can memorize. If you don't want to memorize, that's fine. Okay. My job is not to target you because you didn't memorize those. Okay. I don't know all the properties of all of them. So again, you can break those side chains, those 20 side chains, into 4 different categories. The first one is basic side chains. These side chains usually contain a nitrogen atom or an amino group. All right. We said before the that amino group behaves like a base, okay, because it picks up hydrogen ions in solution. So examples of amino acids with basic side chains are lysine, arginine, and histidine. Okay, and again, right now to us it all means Greek because we're not looking at the detailed structure, properties, and so on. Okay. Then you have acidic side chains, right? These amino acids contain a carboxyl group in the side chain. And examples of acidic side chains are uh, aspartic acid and glutamic acid, right? And here we give you the formula for the side chains. Okay. These are important because they are kind of the intermediate amino acids through which nitrogen moves through living organisms, right? How nitrogen is picked up both by plants and animals. It usually goes through these aspartic acid, glutamic acid, and so on. Okay. Then you have the uncharged polar side chains, right? And these amino acids contain a hydrophilic side chain containing an amino group or a hydroxyl group. And examples of amino acids with uncharged polar side chains are asparagine, glutamine, serine, threonine, and tyrosine. Right? And then non-polar side chains, and these will probably have hydrocarbons, right? So these side chains contain hydrocarbons and are usually hydrophobic or non-polar. And examples of amino acids with non-polar side chains are alanine, valine, leucine, isoleucine, proline, phenylalanine, methionine, tryptophan, glycine, and cysteine. And you can count, there are 20 of them here. Okay. And we are not getting into a detailed discussion of the properties of all these different amino acids, right? How you can go from one to the other and so on. Okay. If you're a medical student, you'll get into that. Our goal is to get some domain knowledge and understand how the quantitative problems that we want to focus on are going to arise, okay, and how we can formulate those. Any questions? And just there's one example, right? One of the amino acids that we listed over there is called cysteine, and this is the structure of cysteine, right? So here you have the amino group on the left, the carboxyl group on the right, and at the bottom you have this side chain which contains sulfur, okay? So in fact, if you have 2 of these cysteines, right, then they can form like between you have SH at the bottom, right? So the SH on another cysteine molecule, right, between the 2 of them, the hydrogen could be eliminated and they could form a bond between the 2 sulfur atoms. That's called a disulfide bond, okay? So, and this can play an important role in determining the 3-dimensional shape of the protein containing these cysteines, okay, because there's this strong bond that is formed. Like it has to be in an environment where, hydrogen is kind of limited, okay. That's when you expect the hydrogen from here to be taken away, hydrogen from the other one to be taken away, and then you have a disulfide bond. Inside the cell, it's an aqueous environment. There's a lot of hydrogen, okay, that's not going to happen. When it's exported from the cell, maybe that kind of bonding will take place. Okay, now the important thing, amino acids can be linked to each other via what are called peptide bonds, which are essentially amide linkages, right? And if you look at this picture, on the left you have an amino acid with side chain R1. On the right you have an amino acid with side chain R2, right? The carboxyl group of the amino acid on the left is going to react with the amino group of the amino acid on the right, right? A molecule of water is going to get eliminated. You can see in the center you do have HH, then OOH. A molecule of water gets eliminated, and you have this linkage right in the middle, which is called a peptide bond. Okay. Remember, I said the condensation reactions are necessary also for forming proteins from amino acids. Right. This is a condensation reaction. Water is being eliminated, right? And now instead of 1 amino acid, you have a protein that is made up of 2 amino acids. Okay. Guess what? Even for this protein with 2 units, okay, there is an amino group on the left and a carboxyl group on the right. So the chain can further elongate, right? Any questions? So let's look at some characteristics of these amino acids. They have a well-defined directionality. You have an amino group on one end and a carboxyl group on the other, right? then it is possible to attach additional amino acids to the existing chain via further condensation reactions, right? And the final chain that results, let's say you've done it 10 times, okay? You have a protein that is 10 amino acids long, but again, you have a polarity. On one side, there'll be the amino terminus, on the other side, you'll have the carboxyl terminus, right? All right, any questions so far? So now if you look at a peptide bond, all right, the 4 atoms that are in that box, right, that is marked in blue, they form a very rigid planar unit. So you cannot do any rotation about the carbon-nitrogen bond. Okay, that is fixed. But the 2 bonds that are linking those alpha carbons, right, the one to the left of that blue box and the one to the right of that blue box, right, there is a lot of flexibility, right? So you can rotate it about that axis, right? So long chains of amino acids are extremely flexible. Okay. So the question is, how will they fold up? Okay. Well, that will depend on the side chains because some of them are hydrophobic, some of them are hydrophilic. Right. So it'll try to fold up into a conformation of lowest energy. Okay. And there are people that study those things. Right. So since polypeptide chains can be of any length and at each location we have 1 out of 20 possible amino acids, there is an enormous variety of proteins that can be synthesized using peptide bonds between amino acids. Okay, and this is an important topic. Like if you see for these proteins, okay, the sequence of amino acids that you have, that is called the primary structure, right? But you would like to predict, okay, based on the primary structure, can I predict the 3-dimensional shape? And that's an open problem, right? If somebody in this class wants to take, that's an open problem. Can you computationally predict that? Okay. If you could, that would be very good because let's say if you want a drug to target a particular molecule, you know what shape that drug molecule has to have, right? So can I reverse engineer that and decide what set of amino acids that I should have, right, to produce that drug, right? That problem is not solved as yet. People are working on it. People in physics, in computer science and all, they're trying to work on that problem, right? But that would be a big breakthrough because if you can predict the structure, then you will know exactly how to produce that drug, okay, or how exactly to target, cure a particular disease. So already, see, it's been like the second lecture and I'm already giving you some open research problems. I'm not working on that, but I know somebody else that is working on it. But I believe that that's an open problem. Am I right? It's a yeah, yeah. So if you can solve that problem, if you have some ideas or something, you'll become famous, all right?
It would like basically make genetic engineering the kind of thing where you're just like, oh, you want something to do
Yeah,
this,
yeah,
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 just
yeah,
look
yeah,
it up. Okay, there you go.
yeah. So you'll become probably a millionaire, yeah, or a billionaire, like if you by licensing that technology. Potential is there. Okay, any questions? So the next set of modular units that we are going to focus on are the nucleotides. These are the units from which DNA and RNA are built, right? Nucleic acids called DNA and RNA are built. So a nucleotide is a molecule that is made up of a nitrogen-containing compound, right, which is called a base that is linked to a 5-carbon sugar. That's why we study 5-carbon sugars, ribose and deoxyribose, right? That carries 1 or more phosphate groups. So we now have all the ingredients, right, to study DNA, okay, to understand what a DNA molecule looks like. Now the nitrogen-containing ring compounds or bases are of 2 types, pyrimidines and purines, okay? So one of them will have a 9-member ring, the other one will have a 6-member ring. Okay. So and I'll give you examples of that. So the nucleotide base categories, if you have a 6-member ring, that molecule is a pyrimidine, right? If you have a 9-member ring, that molecule is a purine. And remember earlier I had said that 1, 2, 3, 4, 5, 6 would get used up somewhere else. So in the sugar you have to number it with 1 prime, 2 prime and so on. So these molecules have already used up the 1, 2, 3, 4. The numbering without the primes has already been used up. Okay, this is how it's going to be numbered, right? So 6-member ring and 9-member ring, and there is a total of 5 molecules like that. Okay, like so you have on the next slide I'll give you specific examples. Some of them are found in DNA. Actually, out of the 5, right, 4 of them are found in DNA, right, and 4 of them are found in RNA, right, but 3 of them are overlapping between the two. Okay. So if you look at pyrimidines, right, Again, these are the 6-member rings, all right, compounds, all right. Uh, you have cytosine, all right, that will have the structure on the left. You'll have uracil that has the structure in the middle, and then thymine, the structure on the right. Again, I'm not asking you to memorize any of these structures. Okay, we want the general properties. Okay. Now notice that thymine is actually 5-methyl uracil, right? If in the uracil molecule, right? See, look at the numbering, right? For the 6-member ring, it's 1, 2, 3, 4, 5. Okay, so you see, so at location number 5, right? You have a methyl group. Remember we discussed methane giving you the methyl group CH3, right, last time? So if at location number 5 you have CH3, right, and in uracil, then you get thymine, right? Now if you look at DNA, you have only C and T, cytosine and thymine. Uracil occurs in RNA, right? It is not found in DNA. Okay. Then you have molecules that are called purines, which have a 9-member ring, right? And the two molecules that you have under this category, they are called adenine and guanine, denoted by ANG. And in easy way, like I used to sit in on this molecular biology class in 2001, and one Aggie pointed out that the easy way to remember is ags are pure. Okay, so adenine and guanine. So you can use that, if you have trouble remembering, just think of it that way. Okay, so these are the purines, right? Any questions? So now we have the structure of phosphate. Okay, we've talked about that inorganic phosphate ion. I've talked about the 5-carbon sugars, all right, ribose, deoxyribose, all right? And now I have talked about these nitrogenous bases, the purines and the pyrimidines, right? So now I'm going to link them together to produce the units that make up DNA and RNA. Okay, so the bases we already considered. Now we also talked about the sugars, right? The two possibilities, ribose, 5-carbon sugar, we've already seen this structure with the 1', 2', 3', 4', and so on. CH2OH sticking out of the plane, okay? And then deoxyribose, where at location number 2', you have removed the oxygen, right? Okay. So now the three different types of ingredients are going to come together to produce one unit of either DNA or RNA, right? So for example, let's say we are working with C, okay? We have A, G, C, T, adenine, guanine, cytosine, and thymine, right? A, G, C, T, and then uracil, okay? We have 5 possibilities, okay? So let's say we are doing this for C. It'll be the same for the other molecules too, for the other nitrogenous bases too, okay? So you have C, you have cytosine on the left, then you have the 5-carbon sugar, in this case it is ribose, right? So what kind of nucleic acid are we going to get out of this? RNA, ribonucleic acid, right? The sugar is ribose, and you have the phosphate, right? So now, see at the bottom of this base, okay, where you see the nitrogen, right? The sugar, the 1' location on the sugar is going to link up over there, okay? Because you do have, at that location, you have OH over there, okay? So probably a water molecule getting eliminated, right? Gets linked up over there, okay? So now you got the base, then you got the sugar ribose, right? And then the phosphate is going to get tagged on, because remember, for the sugar, you had a CH2OH sticking out, right, at the 5' location, right? The phosphate, the inorganic phosphate also has that OH over there, right? So a water molecule is going to get eliminated, and you're going to get a phosphoester bond that is going to form over there, okay? And then these three things linked together, it gives you what is called a nucleotide, okay? So the base linked to the 5-carbon sugar, linked to a phosphate, right? And this is one unit of RNA, right? Units like this will have to be linked together, right, to produce ribonucleic acid, right? Similar kind of units, but with a different sugar, that is deoxyribose, will have to get linked together to produce DNA, or deoxyribonucleic acid. How does it get linked together? Notice that, at the 5' location on the sugar, you have a phosphate, right, inorganic phosphate that is sticking out. At the 3' location on the sugar, you have a hydroxyl group that is sticking out, right? So what could happen is that this is one unit. For the next unit, okay, take the phosphate from the next unit, right? Link that up to the 3' location on the sugar, right? By another phosphoester bond, Okay. Remember, I said it's a phosphodiester bond. Okay, because you already have a phosphoester bond at the top, at the 5' location, right? So at the bottom, you so you already have a phosphoester bond. Each of these units already comes with a phosphoester bond, right? And then you'll produce the other phosphoester bond by taking the phosphate at the 5' location of the next sugar, right? And linking it up with the hydroxyl group at the 3' location of the previous sugar. Okay, so now you have something that is 2 units long, right? Let's say CG. Again, you can do the same thing because you again have a phosphate sticking out at the 5' end and an OH sticking out at the 3' end of the last sugar. Put in another unit, okay? And then that's how you will get a strand of RNA in this case. If the sugar was deoxyribose, all right, then it would be it would be DNA in that case, right? And for DNA, the bases that are used are AGCT, adenine, guanine, cytosine, and thymine. For RNA, it's adenine, guanine, cytosine, and then uracil. Okay. And we will talk more about the detailed structure of DNA and all that. There's a chapter devoted to that. Okay, so I'm not going to spend too much time now, but I just wanted you to finish this class because you told me yesterday, right, or in the last lecture, other than one gentleman here, everybody said they had no background in biology, right? But I want you to walk out today knowing the structure of DNA, right? So that's a big thing, right? I mean, you didn't know, and it's an important thing because we see it all in the news all the time. Okay, so
So just to summarize, DNA or RNA is made up of nucleotide,
nucleotides that are linked together, yes.
and
Bye.
nucleotide is nucleoside with two
Yeah,
phosphates.
nucleoside is just with the sugar, okay? Like if you have a base and then the sugar added onto it, we call that a nucleoside. When the phosphate gets added onto it, it's called a nucleotide.
So nucleotide has two phosphate groups or one phosphate?
Nucleotide has one phosphate.
So you're talking about phosphate group at 3 prime?
No, no, no. See, there, okay, this is one nucleotide, right? So this has got one phosphate group. You have another nucleotide, okay? The second one, let's say this is C, and another one with G, the same kind of thing is there, okay? Now the phosphate on the second one, right, which is at the 5' location, that will link up through another phosphate phosphoester bond with the 3' on this sugar. So now you have 2 units that have been linked up together
So two
by a
nucleotides connected together.
connected together by a phosphodiester bond. Okay, that's how DNA or RNA is formed, and you can really elongate the chain as much as you like. Okay, because if you look at our genes, okay, in the human genome there are like 3 billion of these letters, okay, AGCT, and they're linked together by that phosphodiester bond. And the thing, see, again, the sugar and the phosphate is common between all the units, okay. What differentiates one of these units from the other is what base it has on it, okay. So the sugar phosphate, that forms a backbone, okay. That is not specific to any particular base. But the letters that you have, that C, G, A, T and all that, that basically spells out the biological message in the language of DNA. That has all the information to make you and me just from our DNA. Okay, all the necessary information. Okay, any other questions? So here I'm just summarizing whatever I said earlier. Nucleic acids are formed by stringing nucleotide units together, and I tried to explain how that is done. It will be done in more detail later on. There's a chapter devoted to it. So if the sugar happens to be deoxyribose and the bases that you're using are adenine, guanine, cytosine, and thymine, we have deoxyribonucleic acid or DNA. If the sugar happens to be ribose and the bases are adenine, guanine, cytosine, and uracil, then we have ribonucleic acid or RNA. Okay. Any questions? Now, in addition to forming DNA and RNA, right, these nucleotides, they have other functions, right? They have many other functions. They can serve as storehouses of chemical energy. We discussed that at the beginning of the class, right? Remember I talked about ATP, adenosine triphosphate? That really has that A that I showed you, right? Adenine, guanine, cytosine, and thymine. So adenine is denoted by the letter A. Remember I said A and then there were 3 phosphates, right? Right? So for some reason, again, why did it happen that way? I don't know. Okay, but that's how it's evolved in biology. Okay, because it doesn't look like the nucleotide is necessary for that function, because nucleotide is needed for coding the genetic information. Okay, but in those energy carriers, okay, such as ATP, it's basically adenosine, right, linked together to those 3 phosphates, right? That's adenosine triphosphate. If you break one high energy bond, you get diphosphate. So that's the mechanism. That's the currency of the cell. Okay. So they can serve as storehouses of chemical energy. Then they can also combine with other groups to form activated carrier molecules or coenzymes, right? Like they can combine with other groups to become carriers of energy, okay? Like the energy might be generated in one location, but it's needed somewhere, okay? The cell doesn't have transmission lines, okay? So you can have, you know, some carrier molecules can move out in the cell, they move energy from one area to the other, right? Then they can also be used as specific signaling molecules in the cell, these nucleotides, right? An example is cyclic AMP, right? Or denoted by CAMP. What is AMP again? Anybody remember what is AMP? Monophosphate. So if you have a lot of adenosine monophosphate in the cell, what does that indicate? What is the energy status of the cell? It's low on energy, okay? So that means you have to start burning sugar or doing something to increase the energy, okay? So this molecule, this AMP, right? Yeah. AMP, actually I'll explain this in a moment. Let me just go to the next slide that finishes this chapter and then I'll talk about the cyclic AMP, right? So the recap of this chapter is that macromolecules, polysaccharides, proteins, and nucleic acids found in cells contain a specific sequence of units, sugars, amino acids, and nucleotides. These macromolecules are formed by condensation reactions. I demonstrated that right as I went along, where a new unit is added to a growing chain by the expulsion of a water molecule. The reverse one is hydrolysis. Right? Non-covalent bonds such as hydrogen bonds, hydrophobic forces, etc. make macromolecules assume specific shapes. I said that in the context of proteins. When you're linking up these amino acids together, the side chains have different properties that will determine the final shape, right? And these specific shapes, as well as the non-covalent bonds, allow macromolecules to go and seek out their appropriate partners and undergo the required reactions with them. Just like if two human beings want to interact, okay, you'll recognize the other person just by looking at their face, okay. In biology, it's basically the shape of the molecule. If it has exactly the right shape, then the others there'll be random collisions, okay, nothing will happen. But if it's the right shapes that meet, that's when activity takes place, right? So next time we will go to the chapter about energy considerations in biochemical reactions. But before that, I just wanted to very briefly talk about cyclic AMP, right? So let's just go here. Okay, so you guys told me, right, that adenosine monophosphate is the low energy thing. So you have A, right? You have A here, right? Then see, A is bound to a sugar. Okay, it's not just adenosine. It's bound to a sugar. Let's say like ribose or something like that, right? So you have a structure like this, all right? That's bound here, and then you have a phosphate attached. Okay, so that's adenosine monophosphate, all right? Now here also on this sugar you have 1 prime, 2 prime, 3 prime, all right? And then if it's ribose, I'm sorry, it should have just ended here. The phosphate should have been attached here. So, but anyway, okay. So 4 prime, and then at the 5 prime location you have the phosphate that is attached, right? Right, at the 5 prime location. Now, if it is adenosine monophosphate, okay, if the adenosine monophosphate level in the cell is high, that means the energy level is low, okay. So this molecule could be an indicator of low energy. So cyclic AMP, so this is adenosine monophosphate or AMP. Cyclic AMP means this guy, all right? This guy comes and bonds here, forms the phosphoester bond here. Okay, it's not another unit linking. It's the same molecule AMP, but it's called cyclic AMP because that phosphate links up here. And this is a very strong signaling molecule. Okay, it'll signal to the enzymes, to the machinery. Okay, that enzymes are needed to burn glucose and crank up the energy. Right? It's just like you feel hungry. Okay, if you haven't eaten for a long time, why are you feeling hungry? Because your blood glucose is low, right? So you eat, then your glucose will go up, right? So the cell also has to have a mechanism, right? So this cyclic AMP is a signaling molecule. Again, I mean, why did this molecule have to incorporate a nucleotide? I don't know, right? It could have been something else, but that's how things have evolved, okay? And that's what we have today, right? So the nucleotides are not just important for synthesizing DNA and RNA. they could also play an important role in, or they could serve as a signaling molecule, right? Not to mention their role in being energy carriers and so on, okay, and coenzymes, right? Activated carrier molecules and coenzymes, because it's like charging up a battery that's ready to go, right? If you're looking at ATP, something charged up, you need energy, just break down ATP and get that energy and go from there. So let's stop here for today.